replicated, viral proteins are produced, and then you get assembly of the viral particle, which happens either in the cytoplasm or the ER. And one of the challenges that the virus faces is when the virus pops out of the cell. Remember, the virus has tons of these hemagglutinin molecules, and hemagglutinin binds to sialic acid, which is probably virtually all surface proteins have sialic acid on them on a given cell. And what ends up happening is as the virus pops out, the hemagglutinin binds to sialic acid on the cell and gets stuck. So the virus pops out but gets stuck to the cell surface. Okay, it can't get out. And that's what the neuraminidase molecule does. The neuraminidase molecule cleaves sialic acid residues off of proteins. And so the neuraminidase is required to basically unstick the virus. The virus pops out, it gets stuck to sialic acid on the cell it just already infected and is popping out of. The neuraminidase cleaves those sialic acid residues and allows the virus to be released from the cell. Okay. So the hemagglutinin molecule that we talked about that's important is involved in entry. Neuraminidase molecule is involved in exit of the virus. Okay, so now we'll talk a little bit about epidemics and pandemics of influenza virus. Now, this is characteristic of all RNA viruses. RNA polymerases are about 1,000 to 10,000 times more prone to errors than DNA polymerases are. So all RNA viruses tend to have a high mutation rate because mistakes are made when they replicate. DNA polymerases have proofreading ability, whereas the RNA polymerases don't. Okay. What that means is that every time the virus RNA is copied, mistakes can be introduced. And those mistakes that are introduced can result in changes in amino acids in the viral proteins. If you accumulate enough mutations in that hemagglutinin and neuraminidase molecule, those two molecules that the antibodies in your body target, the virus will no longer be susceptible or be neutralized by your immune system. Okay, and so even though you've got a great adaptive immune response against a particular strain of the flu, as it accumulates mutations, you can become reinfected by a new variation of that virus. Okay, and I want to talk about the terms antigenic drift and antigenic shift as it relates to influenza viruses. So those small changes caused by errors in replication are what's responsible for what we call antigenic drift. Okay. And the idea is that the virus goes in, it replicates, and as it's replicating, there may be a mistake that's made, and the viruses that pop out may contain that mistake, that error. Okay, if you accumulate enough of those errors, you can change the structure of the hemagglutinin and neuraminidase enough that you can become reinfected. So new strains of the virus are produced through errors in replication. So the idea is that people can become reinfected with the influenza virus as it changes. Mutations occur and the virus changes, you become reinfected, and that's how antigenic drift works. That's why you need a flu vaccine every year, because the virus is constantly changing and evolving. Antigenic shift is a much more dramatic change in the virus. An antigenic shift occurs when a new type of hemagglutinin, a new subtype of hemagglutinin, starts circulating in humans, for example. So whereas antigenic drift is the same virus year after year undergoing subtle mutations that cause it to change, antigenic shift results when a completely new hemagglutinin molecule starts circulating in an influenza virus. Okay. And in this case, you have no pre-existing immunity to the hemagglutinin molecule. So even in a seasonal flu, when we have antigenic drift, the subtle changes, you still retain some immunity to the hemagglutinin. The influenza doesn't run that fast, okay? But it changes enough that you can become reinfected. In contrast, an antigenic shift, when a new hemagglutinin molecule comes in, it's, as I said on the previous slide, it's about 30% different than the current circulating hemagglutinins. It's so different that you have no pre-existing immunity to it, and so the infections tend to spread easier, and the symptoms tend to be more severe than in a typical flu. And the primary way in which new strains or new types of hemagglutinin enter into human influenza viruses is through a process called reassortment. And reassortment works because of the segmented nature of the influenza virus genome. So again, most of the viruses we've talked about are non-segmented. They just have a single piece of RNA. But because influenza virus has 8 segments, what can happen is if a single organism becomes infected by 2 different types of influenza virus, if a single cell becomes infected with 2 types of influenza virus, the 8 segments from one virus and the 8 segments from the other virus can kind of mix together. And out of that mixture can come new influenza viruses, which are basically hybrids of the 2 input viruses. So it might have some segments from the human virus and some segments from, let's say, an avian or bird virus. In our modern world, that mixing often occurs in pigs. And there's a couple reasons for that. One, birds and human influenza viruses don't tend to infect each other. So a bird virus doesn't tend to infect a human, and a human influenza virus doesn't tend to infect a bird. Pigs are lucky enough that they can be infected by both types of viruses. So both bird and human viruses are infectable with pigs. And it's actually because although influenza viruses use sialic acid, that carbohydrate, as a receptor, the actual linkage, the way the carbohydrates are linked are important. And the way that they're linked in birds versus humans are different. Okay? Pigs have both types of sialic acid linkages, and so they're infectable by both bird and human influenza. And because of the way we do agriculture these days, we have lots of pigs, oftentimes coming into contact with birds and humans, so they make an ideal mixing vessel for these pandemic or these antigenic shifts in influenza. Okay, and this slide here just kind of shows that bird flus can't get in to a human cell. Human viruses can, but in pigs, if we have 2 different strains of influenza infecting, we can see as you get a mixing of the segments, and out pops a hybrid virus which contains some segments from a bird and some segments from a human virus. Okay, this is a process known as reassortment. And every time there's been a pandemic, there's been a new type of hemagglutinin start circulating in humans. So if we look through the pandemics, the most significant influenza pandemic was the 1918 Spanish flu, which killed anywhere from 20 to 40 million people in a year. Prior to that, the circulating strain was H3N8. In 1918, H1N1, the Spanish flu, started circulating. What's interesting is that, again, you're going from an H3 to an H1 type of flu. And what's interesting is that after that pandemic, the old flu strain is completely lost and the H1N1 becomes the typical seasonal flu. So up until 1957, we had H1N1 was the only type of seasonal flu. Then in 1957, we had a pandemic where it was H2N2, so the H1 was replaced by an H2 influenza virus. In 68, you had the H2 being replaced by the H3 subtype in that pandemic. And in 1977, it wasn't really a pandemic, but you had a reemergence of an H1N1. Okay, so whenever there's been a pandemic, it's because there's a new hemagglutinin molecule that starts circulating in humans.